Good morning, everyone. I am so thrilled to see all of you here today at BIOGAMI Proposers Day. This is an amazing turnout, both in person and online, so thank you all so much for being here. My name is Shannon Green. I'm a program manager in the Health Science Futures Office here at ARPA-H. I've been here about a year. I came over to ARPA-H last January from DARPA, where I'd spent the last decade developing medical countermeasures to help keep the warfighter safe from different chemical and biological threats. And it's really been a fun joy and also a challenge to kind of take off that national security hat and broaden my aperture in terms of thinking about problems that are affecting all Americans. So we're here today to talk about my new program, BIOGAMI, Biomolecular Grammar for Protein Aggregation, Modulation, and Intervention. Now, it's my name up on the slide, it's my name on the website, but this is not just my program. There are a lot of people that I want to make sure to thank up front that help make this program a reality. So my technical team is Julia Gerson, who's sitting up here in the front. Mairead is with family right now, and so she's unable to be here in person, but these two are absolute technical rock stars in helping to craft this program and really give it its meat and bones. So this program would not be here in front of you all today without their amazing hard work. You're going to hear later from Sean Rich. You want to wave Sean for people in the room. Sean is my business financial manager, and she's going to be talking to you guys a little bit later today about how to put together your budgets for a review. And then we also have Danielle Green up in the front, a fellow Green. So we love that. She is our agreements officer, and you're also going to hear from her later today about how to engage with ARPA-H and what different types of agreements with the government look like, and kind of do's and don'ts as well for the proposal review process. So I also want to make sure to really thank as well our AV techs, as well as Renee and all of her crew for helping to put together the space both online and in person. It's really an amazing lift. So before we dive into the meat and potatoes, let's just kind of talk agenda real fast. After some opening remarks from our ARPA-H leadership team, we're going to then dive into the meat and potatoes of the day, where you're going to hear from me a little bit more of an overview of the BIOGAMI program. And we're going to talk about, again, working with ARPA-H and how to put together your proposal budgets. After this monster of a morning session, we're going to have a quick break, and then we're going to come back together for the Q&A panel. So please be mindful of the time, both in person and online, because you don't want to miss the Q&A. This is really your opportunity to get out all those burning questions. You guys have done a phenomenal job of already sending a bunch of questions to the BIOGAMI inbox. If you guys have been tracking online, we've been posting updates to those FAQs usually about 2 or 3 times a week. So please make sure to check for that. You might not see your exact question word for word on that page, but because very often, if you've asked a question, someone else has asked that question too. So we'll kind of collate the language there, make it a little bit more generalizable. But the answers are posted in a very timely fashion up there. Alrighty. So, I know we got some logistics out of the way. I want to pause and kind of take a moment to acknowledge what a special opportunity we have here today, and the opportunity that I have as a program manager at ARPA-H to live every single day. It's not often where you get to come to work with this deep sense of mission and passion. And it's also definitely not taken for granted that when I come into work, every other single person at ARPA-H, whether they're a technical member of my team, whether they're part of the AV crew, whether they're the office leadership, everyone else is driven by that same sense of mission and purpose and urgency. Because we have a really broad mission here, we don't have any time to waste, because the American people are looking to agencies like ARPA-H to develop technologies to our most pressing health challenges. And so when the BIOGAMI program was still a figment of my imagination, still a notion, Dr. Tyler Best, our mission office director for the Health Science Futures Office, was constantly questioning my ideas, challenging me, and also providing that institutional support to bring BIOGAMI to life. So I want to please everyone give a warm welcome to Dr. Tyler Best to talk about ARPA-H and HSF.
And let's see, right here. So before I bore you all to death, PowerPoint by death by PowerPoint, I wanted to maybe open up to, because I can't see everybody online, but by show of hands, how many here in the room have received funding from an ARPA in the past? Okay, so a handful of you all. Could you leave your hands up? For those of you in the room that didn't raise your hand, look around, find someone that you can talk to. These are your mentors for the day to understand what it's like, because it's not like any other funding agency. As Shannon alluded to, we are very much mission driven, deliverables driven, outcomes driven. There are other agencies that are really good at hypothesis testing, doing some of that science that is exploratory or whatever. We are here to engineer, to create knobs on biology that we can turn to manipulate to create outcomes that we want to generate. And the best way to do that is with innovation, with an approach that is much more passionate driven, driven by intrigue, curiosity, innovation, curiosity. I think I said that twice now, but you get the picture. That's what this first image on the death by PowerPoint is. On the far left, you see this bottle with these spikes, wires coming out of it. For any of you that know, just by show of hands, what is that? Sputnik. Okay, yes. So Sputnik in the 50s was a satellite launched by Russia, which was a major surprise to the Americans. President Eisenhower at the time, big kerfuffle in the United States, and so they set up what at the time was called an Advanced Research Projects Agency, because they didn't want to be surprised by new technologies and new developments. Fast forward numbers of years, there's the nodes here in the middle of the picture depicting the first computer-to-computer interaction communication that later became the internet. And the top is the stealth bomber. On the bottom right is a self-driving car, which have become much more ubiquitous and well-known throughout the world. In 2022, President Biden initiated this process for healthcare, and ARPA-H was set up with this Sputnik-like moment of an advanced research projects agency specifically designed for health, so that we could create our next internet of biology of healthcare, or our self-driving I don't know, you name it, our self-driving biology or whatever, that we are able to control, our stealth systems that we can generate so that we have the health outcomes that we want. So this is in effect a Sputnik moment that Shannon has created for these IDPs, misfolded proteins, that we can go after some of these diseases and disorders that for decades people have been banging their heads against and not finding progress and not advancing or creating solutions for. And so this is your opportunity to think beyond that next incremental step of science. Most of us are scientists, if not, engineers or whatever, but science is designed around changing one variable at a time and testing the results, change another variable and test results. That's great science, and we want you to do that and have proper controls and everything. But your approach to solving this problem should not be incremental. We shouldn't expect proposals from you that are just changing that next variable. We want paradigm shifts where you're changing multiple variables. You're looking at the problem that Shannon has outlined on these IDPs in a much different way. Be creative. This is your chance for you to propose those things that you have been thinking about in the back of your head for a number of years, or you and colleagues have talked about at a conference, at a bar, wherever. You're like, oh, that would be cool if we could do this. But then you're like, yeah, no, there's no reality. We can't do that. This is your chance to do those things, to try the innovation, to try those things that you've been thinking about, that have been driving you forward, and you just never have that chance. This doesn't mean we're looking for tinfoil hats, but excite Shannon and her team, excite her with the innovation that you're proposing, the creativity and the curiosity in which you're coming about trying to solve this problem. base it in reality, base it in physics, base it in chemistry and facts, but be creative. This is your chance to really wow her. A couple other notes before death by PowerPoint. She, as the program manager, she is going to be your strongest advocate. You need to convince her that what you're doing is the greatest thing since frosting on sliced bread. She's also going to be your harshest critic. If you're not performing, if you're not doing good science, if you're not doing the things that you said you're going to do, she has the ability to negotiate an end to your contract. And basically you're done, and you get to move on to other things. But she's going to be your greatest advocate to potentially get more funding for more innovation, more projects, or your greatest critic. And yeah, I think those were my main points here. The last thing is why are you all here, either online or in person? I think the elephant in the room is you want money to do research. Yes. But to Shannon's point, we're mission driven. There are people out there. I had a grandfather. We all have family members who are suffering from either neurodegenerative diseases or other diseases where protein misfolding is a major cause or a correlative impact of those diseases. It's those people that we want to help. The other two main reasons you're here is for teaming. You can't do this alone. You need to find the partners in which you can collaborate with them and get this done. If you've ever tried to lift a piano, you can't do it by yourself. You need a team. You need someone that knows how to do it. You need the people that have strong backs, that have the drive to really push on when it gets heavy over time. And then the third reason, obviously, is to hear from Shannon and get a better understanding of what is meant, what is written in the ISO. I'm sure you have all read it 20 times and have it memorized, diagrams on your whiteboard on how you're going to address every issue, every should, every must in that ISO in your proposal. But there are some nuances that could be left to interpretation, and that's why you're here, to get those answers out of Shannon and her team. The rest is just slides. How ARPA works, who we are, what we do, and what we are wanting. We want radical change, autonomy. We come in on term limits. Shannon is, as she mentioned, she started here last January. That means she's a year plus a month into her 3-year term. We can be renewed for another 3-year term, but that's it. I am myself, I'm coming towards the end of my 3-year term. There's always technical, scientific change and overlap. So we get fresh ideas. We have different perspectives on the stuff that you're proposing now. There may be a different program manager 3-4 years from now that is going to look at it in a different way. And you need to make sure that she has advocated sufficiently for you so that when that transition happens, you're still moving forward and progressing. Our ecosystem, Shannon also alluded to it as to why she came from DARPA over here to ARPA-H, is everybody. Everybody is affected by healthcare and needs healthcare. And that is our ecosystem, whether it's the nonprofits, the patient advocacy groups, other government partners, academic institutions, et cetera. We want to work with them. We want to serve them. We have clients, we have performers, we have customers. If you're familiar with an ARPA like DARPA, IARPA, we kind of follow a similar process where a program manager comes in. They really dig deep on identifying a problem, formulate a plan around that problem, send out the solicitation. That's where you're at now. Move on those solicitations, receive proposals, evaluate, make those awards, and then the bulk of the work is program management. She's going to manage your effort. These are not grants. You're not getting money thrown over the wall to you to play with. Shannon is going to be on you every month or more often than that, bi-weekly, weekly, checking in on you, making sure that you're delivering on your milestones, your metrics to get this done. And that's it. And then you meet, You generate a product, a proof of principle, a concept that then is transitioned and is going to impact and create healthcare for others. Not all of you are going to succeed. Alluded to this earlier, like you're going to give it a try. Let's take on those risks, and maybe that's why you're here, is to try something really, really risky. And if it doesn't work, that's great. You tried it. And Shannon will negotiate with you to end that contract early so you can move on to the next thing. If, however, biology never goes according to plan, but it may go according to with the plans, and you're successful, and you move throughout the lifetime of the program, and you continue on and meeting those milestones, and you keep progressing and succeeding. Those are the kinds of depictions here on this chart on the performers. All right. A little bit of organization within ARPA-H, and this is a little bit of a black box to many of you. We're working on better communication here on how we are organized, but we have primarily right now 4 mission offices or areas of research that we fund. Shannon, I lead the Health Science Futures office right now. Shannon is one of the program managers there. We are really focused on the future of health. We feel that the best way to predict the future is to control it, and therefore we want to be able to analyze, diagnose biology, and then therefore also be able to create or control it so that we can generate the health outcomes that we want. In effect, kind of a read and a write on biology to generate those health outcomes. BIOGAMI is a really good example of that, where we're trying to predict and understand protein folding, misfolding, all those interactions and how they are affecting, so that we can then control those proteins in a way that's going to impact our health. Other offices, like the Scalable Solutions office, is really focused on taking technologies, proofs of principle, and scaling them up and out so that they get into the hands of everyone, overcoming geography, overcoming costs, so that everybody can have them. Our Proactive Health office is really focused on making sure that we don't get sick. So it's a little bit of preventive medicine, but along the lines of all sorts of aspects of making sure that as we age or as we grow or as we're exposed to things, that we don't get sick, as a way to reduce healthcare costs for the nation. The Resilient Systems office is focused much more on systems in general, not biological systems per se, but the systems that surround healthcare, hospital systems, robustness to infrastructure, cybersecurity, et cetera, that surrounds that. All right. And as an example, my last slide before you all fall asleep is a list of some of the programs that we have in Health Science Futures that exemplifies why and how we think. We have the Nitro program, looking to replace or make obsolete those invasive surgeries to replace a joint with injectables. A Psi program that is looking to image and be able to image cancer in the surgical suite, so you remove all the cancer tissue at once. You don't have to go in for repeat surgeries to do that. The Embody program is looking at in vivo cell therapies. Instead of taking all the cells for cell therapies out, transfecting them, changing their genetics for specific targeting, do all that in vivo rather than ex vivo, and all that process. Other examples, AIR just launched in late December is looking at autonomous robotics for endovasculature or novel ways of creating little microbots, et cetera, et cetera. You can go on the webpage and read more about all those. With that, let's actually dig into BIOGAMI and find out what Shanna actually wants.
Awesome. Thanks, Tyler. Okay, so let's dive into BIOGAMI, which is really honestly why we're all here. So as Tyler mentioned, we all probably have someone in our lives that is affected by a disease revolving around protein misfolding and aggregation. 3 out of my 4 grandparents had dementias and various forms of neurodegenerative disease. I also lost a partner to suicide following a traumatic brain injury. So this really hits close to home. And when I think about the future, what I want to imagine is, what if you could maybe take a daily pill that could prevent the proteins in your brain from misfolding? So we don't have to imagine a future where neurodegenerative disease is hanging over our heads. What if we could detect and repair those earliest stages of protein dysfunction to eliminate neurodegenerative disease? BIOGAMI aims to build the next generation AI and experimental models for intrinsically disordered proteins and aggregation. We want to leverage those cutting-edge tools to develop novel therapeutic modalities that can interfere with or prevent IDP misfolding and aggregation while preserving those critical protein functions, and also create novel detection capabilities that respond to very early stages of protein aggregation. If we are successful in this program in modulating the dynamics and the aggregation of intrinsically disordered proteins for neurodegenerative disease, ARPA-H is going to be able to propel early intervention modalities for other types of chronic diseases and beyond. So, why do we care? I feel like this slide kind of speaks to it better than words really can. We care because there are millions of Americans that are affected by diseases of protein misfolding and aggregation. And we typically think of these diseases in the neurodegenerative realm, right? Including Alzheimer's, Parkinson's, and ALS, and others. But protein misfolding and aggregation play a role in chronic diseases as well, including cancer, type 2 diabetes, and even latent viral infections. The impact of a new therapeutic paradigm for the treatment of neurodegenerative disease is immense. Alzheimer's is the #1 driver of health costs annually, with $360 billion spent on treatment alone in 2024, and these costs are expected to rise astronomically in the future. But even less prevalent neurodegenerative diseases like ALS have a very outsized economic impact, and ALS is actually twice as expensive to treat than heart disease or cancer per patient. So if we can have a dramatically new approach to treating these types of diseases, this will have far-reaching benefits, not only just in patient care and direct cost savings, but also thinking about that 70% of dementia care costs are currently borne by families and unpaid caregivers. And most critically, up to 80% of proteins implicated in disease states are currently considered undruggable because of their inherent disorder. So by developing tools to address upstream protein misfolding and aggregation, we could prevent enormous downstream costs and impacts to quality of life. So I think I'm probably preaching to the choir here, because if you're here in the BIOGAMI Proposers Day, I'm probably pretty familiar with intrinsically disordered proteins. But these are proteins that contain domains called intrinsically disordered regions, or IDRs. And these are defined by the lack of a stable three-dimensional structure. Because of their unstructured nature, IDR domains are highly responsive to diverse physical and chemical stimuli. These types of proteins are able to self-organize into biomolecular condensates, which is great because it enables the execution of essential biochemical reactions inside the cells in a very localized manner. Now, these IDR-containing proteins are incredibly prevalent. 70% of proteins in the human proteome contain at least one IDR of 30 amino acids or more. And these IDR domains can be found in different parts of the proteins. They can be found as flexible linkers between more traditional folded domains. They can enable the protein to adopt different types of conformations, to do different types of jobs in the cell. However, these highly flexible proteins, being able to do a lot of different types of jobs in the cell and being able to respond to different types of stimuli, are incredibly useful, but also very risky, because when things go awry with these proteins, the impacts can be multiplied, leading to protein aggregation and disease pathology. Neurodegenerative diseases, as I mentioned, are prime examples of the ways that intrinsically disordered protein misfolding can lead to negative health impacts. Neurodegenerative disease-linked proteins are often associated with toxic intermediate species of pathological aggregates, including those of FUS, TDP-43, alpha-synuclein, and tau. And pathological aggregates are observed in brain dissections and in vitro when seeded with pathological proteins. Protein variants, including isoforms, mutant sequences, and aberrant post-translational modifications, can shift these proteins between more dynamic and liquid-like states towards more gel and solid-like states, and bias dynamics towards aggregation. Despite multiple potential triggering events, including inflammation, vascular dysfunction, DNA damage, and others, And a common failure point for degenerative disease is the protein misfolding and aggregation. So the problem that we faced here today, though, is that we know that misfolding and aggregation of intrinsically disordered proteins can lead to disease, but intrinsically disordered proteins as a whole are very poorly understood. And our current lack of understanding of the dynamics of these proteins and what triggers those initial misfolding events limits our ability to combat diverse diseases and closes the door to new therapeutics. What we really need and what we're aiming to do in this program is develop a radical new way of modeling how disordered proteins move between different shapes, how they phase separate within cells, and what types of mistakes can lead to pathological and toxic aggregates so they can work backwards to a new upstream points of intervention. The BIOGAMI program is going to do just that. We're going to develop this comprehensive suite of computational and experimental platforms for disordered proteins that will enable us to reliably modulate how proteins fold to both reverse and prevent misfolding and aggregation, and also enable highly accurate early stage predictors of protein misfolding. My goal for this program writ large is to enable a toolkit that can take any intrinsically disordered protein, be able to model and predict and experimentally validate how it moves through different states in different chemical compositions, different mutations, different post-translational modifications, so that we can highlight where things are going to go wrong and develop interventions to prevent that. So what do we need to know in order to control protein folding and condensate behavior? The first part of this program is Technical Area 1, which is building an understanding of the molecular grammar that governs IDR folding and aggregation, and being able to accurately predict and model the structure and function of these proteins in both healthy and disease states. Next, if we are able to have these capabilities, both these computational and experimental tools, then we can leverage those to develop both indicators for early detection of protein misfolding, as well as developing modulators of protein misfolding and aggregation. So as I mentioned, and if you've read the ISO, and I hope all of you have, BIOGAMI is organized into two different technical areas, and proposers need to address both TAs in successful proposals. I'm going to walk through details of each of these technical areas in subsequent slides, but at a high level here, TA1 is going to work to establish that molecular grammar of intrinsically disordered proteins, again, that foundational knowledge upon which TA2 is built. And so leveraging those TA1 models, TA2 is going to develop technologies to detect and prevent protein misfolding. BIOGAMI is a 4-year program. So as Tyler mentioned, I'm on a clock. I've already spent 1 year at ARPA-H. My 3-year term, I have 2 years left. Hopefully I'll get renewed for another few years. This program might potentially outlive me. So what I hope to do as a PM is select the best proposals that are going to develop the technologies that are going to live even beyond my tenure at ARPA-H and are really going to transform how we think about degenerative disease. So proposers are going to work on both TA1 and TA2 in both phases of the program. Phase 1 is much more heavily weighted towards TA1 platform development with some initial proof of concept TA2 work that's going to start adapting those TA1 high throughput experimental models for disease indications. We're going to talk more about those in a second because I know you guys have had a lot of questions about that. In phase 1 with TA2, what we hope to develop are early stage binders. Those can be small molecules, peptides, larger biologics that are able to interact with early stages of misfolding of those IDP proteins. In phase 2, we're transitioning much more into validation in disease indications and turning those early binders of disordered proteins into modulators that see if they can actually prevent protein aggregation, as well as serve as a readout of early stages of misfolding and aggregation, and actually turn those early leads into actual assets for clinical development. BIOGAMI's end goal is therapeutics and early aggregation biomarkers. And so as such, reading the ISO, we're requiring performer teams to include what we're calling a translation advisory board, basically scientific and commercial input that will help make sure that we're not losing sight of that end goal, that this doesn't become just a science project. Again, we're not NIH. We're not going to send out a grant where we give money and say, go forth and do good science and publish some science and nature papers in a few years. That's not how we work. Our end stage are those early clinical assets. And so we want to be keeping that in mind, even though those aren't deliverables until the end of year 4. We need to be continually driving towards that and thinking about what is the translation of this technology post ARPA-H. Because again, I'm not going to be around forever, and the BIOGAMI program is also going to end. So we want to make sure that your technologies are going to live on, that they're going to translate towards commercial success and clinical success. All right. So let's dive in now into the different technical areas. So TA1, again, is establishing the molecular grammar of disordered proteins. We know that this is a big challenge. There has been a lot of really exciting work in the last couple of years. I swear I can't open my phone without seeing new papers being published about new models of disordered proteins or predictors of aggregation. And these are all really exciting findings. But where I think we're falling short for an overall goal of what I have in my brain and what I want y'all to make into reality are these experimental platforms that would enable us in a very high throughput and fast manner to validate those predictions. Because what I've seen so far, and it's really, really exciting work, like y'all are on the cutting edge, is looking at short sequences of disorder that are some derived from natural proteins and some are synthetic sequences. But what we need to look at is the disorder in the context of full-length proteins where you might also have folded domains. We also need systems for being able to assess different types of mutations and post-translational modifications, and actually being able to validate and really being able to understand what's going on in vivo. One of the other key milestones in TA1 is being able to look at the interactome of these disordered proteins. There's a lot of hypotheses as to the impacts of aggregation in disease. There are some hypotheses that say that if your protein is misfolding and aggregating, you're getting a loss of function because it's no longer able to interact with other cellular partners in ways that it would normally do in a healthy state. But those aggregated proteins can also have a gain of function in which those aggregates are able to interact with other cellular partners. And so we want a capability to be able to understand both that loss of function from a healthy state and what protein interactions are we now missing because of the aggregation, and then what also are we gaining. So requirements for TA1 are to develop both test tube as well as cell-based high-throughput experimental screening platforms to be able to assess aggregation, phase separation, condensate formation, and IDR functions and interactions across a diverse IDR sequence space. And then leverage those screening platforms to be able to establish computational models that can then help us to predict with novel IDR sequences or other proteins that we haven't previously trained on, to be able to define functions of those IDR-containing proteins and enable this drug discovery pipeline to target aberrant protein aberrant behavior. So the proposals, and Tyler mentioned this as before, in the ISO, you will notice there are words that say proposals must, proposals shall. I would highly recommend that you guys take the ISO and do a control find function and do a search for those words and make sure that you're looking for every instance of a must or a shall, because if those details are missing in a proposal submission, it could be considered non-conforming. So really want to make sure that you're reading in great depth and looking for all those details that we're requiring. So proposals must include detailed descriptions of the computational models, the types of training data that you're going to use for those models, pre-processing, quality control, et cetera, et cetera. For the in vitro and in vivo models, please provide rationales, again, for the different types of model systems that you're proposing, details on assay robustness, sensitivity, dynamic range, and alternative approaches. We also want to understand what's the model throughput, scaling, and speed, what it can be on day one, and what you're aiming for at the end of the program. We also want to understand the breadth and depth of sequence diversity. And we had a question in FAQ about this, and happy to address it more in the Q&A panel. This is really kind of up to you guys how you want to approach this question. Do you want to start with a very broad number of sequences and more shallowly explore mutational space? or start with a smaller number of IDR sequences to train your computational models and really explore the entire mutational space of those different proteins. I don't have an answer. This is where proposals come in, and we want you to defend your choice. And then also the proposals need to include a strategy for how you're going to validate these foundational model predictions. Then TA2 is how do we take this foundational molecular grammar and actually translate it into assets for clinical development. So TA2 is aimed at, once we have this understanding of the behavior and the dynamics and the kinetics of protein misfolding and aggregation, can we modulate that behavior to prevent aggregation from occurring in the first place? So key milestones for TA2 might be to have to adapt TA1 model experimental systems for your protein indications of choice for the diseases that you're interested in, right? We're going to require you to identify candidate indicators for early detection of protein misfolding and aggregation, as well as candidate molecules that can prevent or reverse protein aggregation, and then demonstrate prophylactic or therapeutic efficacy in animal models. So proposers must develop indicators of protein misfolding and aggregation against 2 indications. So they have to fall also into 2 different categories. So neurodegenerative disease and a non-neurodegenerative chronic illness or other peripheral protein misfolding disorder. Now, one of these diseases needs to be rare and one needs to be more common. So this could be divided into one rare neurodegenerative disease and one more common non-neuro chronic illness, or a more common neurodegenerative disease and a more rare peripheral protein folding disorder. Proposers need to, we're not being prescriptive and telling you what diseases we want you to work on. This is up to you guys. So if there are diseases for which you already have robust preclinical models and you want to work on that, that is fantastic. If you want to build out a new model system, that's also fantastic. We want you just to defend your choice as to why you want to go after these different indications. In phase 2, we're going to then refine the early stage candidates and really focus in on the neurodegenerative indication. And we'll talk about this a little bit more with Charlotte in a minute. But I want this program to focus on the hardest of the hard. So neurodegenerative disease is really freaking hard. I'm just going to put that bluntly. The models don't always recapitulate human disease. They're long, they're expensive. Getting therapies across the blood-brain barrier is also really difficult. But we wouldn't be an ARPA-style agency if we didn't go after the hardest of the hard. So we're setting that benchmark that you're going to start developing your indicators and your modulators against those 2 different diseases, but that we're going to focus most heavily in phase 2 on neurodegeneration, because if we can crack that nut, then developing modulators for other types of diseases that are more accessible than having to cross the blood-brain barrier should be a lot easier. So we're really putting it to you guys in terms of these metrics, but I think y'all can handle it. I'm really excited to read all of the proposals. There are details for TA2. Proposals must include descriptions, again, of the health and/or economic impacts of the chosen disease indications. We want to see both the qualitative and quantitative metrics for detecting the protein misfolding states, as well as functions and interactions. We also want to understand how you're going to look at detecting off-target effects of these different protein folding modulators, and then how you might use biorepository samples to try to assess whether your indicators of protein misfolding might be detected in human tissues. Okay, so program logistics. We are here. We're February 20th and 2026. Solution summaries are due on March 4th. I know that's not a long runway. I understand that. But that is why we are here today. This is why you guys are here in the room. This is why we've allocated like 3+ hours to lightning talks where you guys can hear from each other, also mix and mingle during the breaks, during lunch, talk to one another, and form those teams. like all ARPA-H programs, they are very multidisciplinary. It is, I don't want to say never because I'm a scientist and I always like to hedge my bets, but it is highly unlikely that one single lab is going to be able to accomplish all of the BIOGAMI goals. It's highly likely that we're going to need to partner and have diverse expertise. That's not to say that it couldn't all be found within one institution, but teaming is going to be your best bet for being able to propose successfully to this program. As I mentioned before, read the ISO carefully. Again, do that search function for proposers must, proposers shall. Make sure that proposals are hitting all of those points. Again, our FAQ is still open. Please email all your questions to the BIOGAMI email address. Don't send them to me or my team because we're just going to send an email right back to you and tell you to send them to the BIOGAMI inbox. So please just go ahead and do that. Again, we are being very timely with how we're updating our FAQs. We're trying to get to those like 2 to 3 times a week. So your questions should be answered up there pretty quickly. Full proposals are due on tax day, April 15th. So just like the IRS, we also don't take late submissions. So please make sure that you're familiar with our submission portal ahead of time. Don't try to log in 5 minutes before proposals are due and then come send a sad email and say, my dog ate my homework and I really tried to submit it on time. We're going to say we're sorry, but we told you at Proposers Day. Remember when Shannon said to log in early and make sure you know how the system works? Please do that ahead of time so that your proposal gets reviewed. In between solution summaries and full proposals, we will be reviewing those solution summaries and we'll be providing feedback to you all in a very timely manner, again, because we recognize March 4th to April 15th is also a pretty quick turnaround. So we are going to turn around feedback to you all very quickly, either encouraging or discouraging a full proposal. This feedback is not binding. However, if you do receive feedback that a full proposal has been discouraged, it's highly unlikely that a full proposal would be found selectable for funding. So please take that feedback to heart. Let's see. I think those are the main points that I wanted to hit here. These slides are going to be posted to the BIOGAMI webpage. And again, a recording of the webinar is also going to be posted to the BIOGAMI webpage. So don't feel like you have to frantically take notes. These are just some of the glossaries because we're government and we like to use a lot of acronyms. So this could just be helpful. Let's see. Sean, how are we doing on time? Perfect. All right. So what I would like to do now is actually pivot from talking about the nuts and the bolts of BIOGAMI and actually take a step back to look at the bigger picture. So if we are successful in this program, we can imagine a future where no disordered protein is considered undruggable, where we could take that daily pill to prevent proteins from misfolding and aggregating. But recognize that there's a big gap between this early stage, high risk research that we're taking on in the BIOGAMI program and actually making clinical impact. And so to talk about the translational investment landscape, I'm going to invite Charlotte Hebert, who's joining us virtually to talk a little bit about how to attract venture funding and how to move BIOGAMI technology from the bench to the bedside. So Charlotte joins us as a vice president of corporate development from Lineage. She started there in April 2024. She has over 15 years of basic science research background and over 15 years in biotech venture investment experience across a broad range of sectors, therapeutic modalities, and stages. She's also strategic director at Madrona Venture Group, where she advises their growing AI/ML driven life science portfolio and serves on the board of directors of the Beckman Research Institute at the City of Hope. Charlotte is our commercialization mentor for the BIOGAMI program. And if you are a selected performer on the BIOGAMI effort, she is going to be instrumental in helping to engage with the VC and the pharma communities in order to ensure that our technologies make it from the bench to the bedside. So, Charlotte, over to you.
Thanks so much for that great walkthrough and the introduction. So one of the things that's really exciting about the BIOGAMI program is that we've never known more about the biology of neurodegenerative disease, but understood less about how to intervene. And I think that one of the things that's most interesting about taking basic science and making a product or a patient intervention in the world is the role of those risk governmental dollars in getting the ball rolling. For this program, I think we thought very carefully about the endpoints that would be most interesting for additional investment. And it's something to keep in mind as you're thinking about designing your programs as an individual or as a team. One of the things that we'd love to do is really identify sustainable solutions that create a data package that is amenable to commercial interest and venture investing. Despite any macro environmental news you may read, it is still a very good time to be in the business of innovation. This, I think, very profoundly interesting and impactful area of neurodegenerative disease has never enjoyed more interest and focus from a venture investing group to the larger biopharma and biotech community. So from private investing to public companies, there is an appetite to address these markets and a recognition that there are fundamental changes and insights into the biology of how to intervene with, prevent, or reverse misfolded proteins. One of the things that's fantastic about those commercial interests is they're often PhD level scientists like you or I on the phone and really understand, I think, the insights that help us identify a science that can become a technological intervention or a product. You've probably run into this at your tech transfer office as you file intellectual property, as you think about taking your ideas into the world. They have great resources as well. And so you can think of the feedback that we're going to give you as sort of like in addition to when you think about, what you can do with your local tech transfer office. One of the most important things, I think, is the breadth of networks we have to bring to bear to, I think, help you think about endpoints that are really going to be durable and of commercial interest. So we have networks that we can get to in the venture capital world, some of the most aggressive investment dollars that are looking to make fundamental game-changing investments in new and emerging technologies. There are folks who have made their entire careers around identifying technologies that are high risk but high reward. And those networks are already on the phone today, listening to these proposers today, and certainly available more broadly as BIOGAMI becomes public. I think it's also important to remember that we may make research products as well as patient therapeutics along the way of addressing all the phases of biogamy. And there never has been, I think, more appetite for new tools investments. So you can think about the larger companies that Thermo Fisher's and others like as actually being quite involved in staying up on new science, as well as watching those investment dollars that are trying to take basic research and make them tools. In addition, there's also the classic licensing models that you're familiar with when you think about being able to interact with a larger multinational company. And lastly, having a preponderance of data, evidence, and insights to actually create your own company. So these are all things that I've been involved in before. And so we can give you durable feedback. And if I don't know it, connect you with, literally a huge network of advisors that understand this landscape and have stood in your shoes before. I think one of the things that's most interesting to remember is it's never too early to think about these considerations about what makes sense and what would be investable. We don't expect you to have this at your fingertips that already, but we do want to provide that guidance as you propose and develop your ideas to make sure that they're in alignment with the dollars that are out there. I think the last thing to really think about is at the end of the day, sometimes as scientists we think a little bit small and focused, and I would encourage everyone that think bigger. and think riskier, because these are the dollars that take the risk to get the data that will attract folks who can take these sustainable solutions beyond ARPA-H dollars and actually spend other people's money later on to help make these products and interventions available for patients. So I know it's a very interesting time for biotech and pharma. We are now, to my view, a permanent part of sort of the market landscape, as well as, public investing and IPOs are still on the run. So we are watching those trends in investment very carefully and thinking about how we can connect each of these different proposals that get funded into these arenas and make them, I think, real objects of investability in the world. So it's the best time to dream about what that invention can be, and get the best time to get our durable feedback of how that could be aligned with current companies that are out there, as well as the creation of new codes. So with that, Shannon, sort of like given a really what I hope is a positive spin on how each of your ideas really are going to be under review for becoming a product, as well as having these networks available to help you progress along the way and actually make a continuity handoff into other dollars that can help you move forward.
Fantastic. Thank you so much, Charlotte. Okay, so we are going to pivot, talked a lot about BIOGAMI, the program as a whole, investment future, kind of where we want to go with these assets, but how do we actually get there? How do you actually work with ARPA-H? What does the solicitation proposal review process, contracting process look like? So I'm going to invite Danielle Green to come up.
Hi, everyone. As Shannon introduced, I am Danielle Green. I'm an agreements officer with the ARPA-H Business Innovation Division supporting the Health Science Futures Office. And I'm going to be talking to you about the Innovative Solutions opening. So just to start with some ISO basics, ISO is short for Innovative Solutions Opening, which is ARPA-H's term for solicitation. It's, similar to what you would expect a broad agency announcement to kind of mirror. So ISOs are designed to enable merit-based awards, which allow the government to focus on the technical quality of the submissions rather than just the price. This means that we're looking at the best ideas, not necessarily the lowest cost. Rather than prescribing a single solution, ISOs encourage a wide variety of approaches to solving problems, and then they also offer flexibility in the types of award instruments that can be considered. So some key highlights of the BIOGAMI ISO is that all of the awards will be made as other transactions, and all types of entities and teaming arrangements are encouraged to participate. There are no funding limits for individual projects. We do strongly encourage questions, and answers to the questions are posted publicly on the FAQ program page. We also post to sam.gov, but rather than posting the FAQs bi-weekly, we just posted the link to the program page on that SAM posting. Solution summaries are mandatory, and you may submit multiple solution summaries, but you may only submit one proposal per team, only after submitting a solution summary. If you are proposing, if you're a member who's proposing on multiple teams, ARPA-H may want to establish an associate contractor agreement. It's also known as an ACA. If you're not familiar, these are just formal agreements that set terms for how contractors will cooperate with one another, sharing data, resources, but protecting any proprietary information. ACAs also help teams coordinate, share information, and avoid duplicating efforts across related projects. All right, so moving on, as Shannon had mentioned, a submission can be deemed non-conforming. That means that a non-conforming submission may not be evaluated, but as we said, you'll be informed of every step of the process. And then you can always reach out to our email, BIOGAMI at ARPA-H.gov with any questions. So what are other transactions? What we like to call OTs. They are flexible, collaborative, and legally binding agreements that are not traditional procurement contracts, which are generally subject to the Federal Acquisition Regulations, known as the FAR. They are also not financial assistance awards, so they are not grants and cooperative agreements. So OTs are modeled after more closely on commercial contracting. They're, like I said, not subject to all of the federal regulations, and this allows for more open negotiations and greater partnership between the government and proposers, making it easier to share resources and knowledge. And for your reference, we do have an ARPA-H sample OT on our ARPA-H website under the Other Transaction Community resources, and we do show how to get there. Let's see. There's a QR code for how to get there. Okay. All right. So who can apply to the ISO? All responsible sources that can meet the government's needs are eligible, including commercial organizations, academia, nonprofits, small businesses, and ARPA-H can make multiple awards to a single entity. Federally funded research and development centers and other government entities are generally not eligible to participate, but you can reach out to the BIOGAMI email if you have questions. If you think you have a unique capability, we will talk to you. However, like I said, generally not eligible. Also, if you currently provide contracted support to ARPA-H, you are ineligible due to a conflict of interest. Regarding awards, priority is given to U.S.-based entities. However, non-U.S.-based entities can participate as long as they're compliant with U.S. laws and regulations. There are certain foreign entities and individuals that may not participate and are excluded, as listed in the slides here. And then finally, all proposers must have that active SAM.gov registration throughout the process. Okay, so how to submit. First, you have to submit a solution summary. This is mandatory. It's limited to no more than 5 pages, and you have to follow the guidance in the Appendix A of the ISO. You'll submit it through the ARPA-H submission portal, and you will receive feedback. And then as long as you submit a solution summary, you can submit a full proposal. This will follow the guidance of Appendix B of the ISO, and it also is submitted through the solution summaries or through the submissions portal. All right. Next, we'll talk about how these submissions are reviewed. Solution summaries are independently reviewed, and only within the ISOs, those that are within the ISO scope will move forward. Again, you'll receive feedback indicating whether or not you're encouraged or discouraged from submitting a proposal. However, you can submit a proposal regardless of the feedback you receive. Like Shannon said, if you are discouraged from submitting a proposal, it's more likely that your proposal will not be selectable. Full proposals are also independently reviewed. They are evaluated against the evaluation criteria outlined in the ISO. And it is noted that proposals submitted are not evaluated against one another. You'll be notified again at each step of the way in the process, and all communication regarding your submissions will be sent through that ARPA-H solution submissions portal. All right. When it comes to how we evaluate, there are 4 main criteria that we'll be reviewing your approach against, and that's in descending order of importance. First is overall scientific and technical merit. So the reviewers will look for evidence that your approach is innovative, feasible, complete, and with clear deliverables and risk mitigation strategies. Second is your team's capabilities and experience. Reviewers will look for evidence that your team has the skills and background needed to accomplish the proposed work within the budget and schedule, and including demonstrating capabilities in similar efforts with clear descriptions on the key personnel's qualifications. And then if your team does have related government project experience, be sure to describe that as well. Third is the potential contribution to the ARPA-H mission and the user experience. So reviewers will assess how your proposal advances the ARPA-H mission, and then also how your solution benefits the end users. They'll look for projects that address unmet needs, have the potential to improve health outcomes, and have the potential to transform biomedicine. They're also interested in whether your work encourages future research, commercial or clinical applications, and if you're taking an interdisciplinary approach to solving the problem. And the fourth and final criteria is the assessment of the proposed cost and price, ensuring that it's reasonable and aligned with the technical goals of your work. Cost realism analysis may also be conducted to ensure that the costs are justified. And also proposals that offer resource sharing. So that's things such as leveraging existing assets, resources, or collaborating with partners. Those are viewed positively, and especially if they enhance the project's commercial potential. All right. So to wrap this up, keep monitoring sam.gov for any ISO updates. Remember that only formal amendments that are posted to sam.gov can change the terms of the ISO. Make sure that if you're not already registered in sam.gov or in the process of obtaining an active registration, that you register as soon as possible. Make sure that your submissions conform to all of the ISO requirements, meaning carefully read through the ISO, all of its appendices, again, because non-conforming submissions may be rejected without review. Make sure to note the key deadlines for solution summary and full proposal submissions and read the ISO and the FAQs before submitting questions. A lot of the time we'll get a question, but it's already answered in the FAQs. Regarding questions, make sure that all of your questions are as clear as possible and that there's no proprietary information included and that they're sent to the BIOGAMI email. And also, if there's any contradiction between what the ISO says and what the FAQs say, always defer to the ISO, but go ahead and just let us know if you find a contradiction somewhere and we'll go ahead and address that. Other than that, that is my briefing on the ISO. Thank you.
Okay, good morning, everyone. My name is Sean Rich, and I'm Shannon's Business Financial Manager. In the next few minutes, I wanted to talk about a quick overview of the cost and administrative requirements for BIOGAMI. One important note that I wanted to make up front is that if you're selected for award, having a clear, compliant, well-supported cost proposal can significantly reduce the back and forth during negotiations and help us move more quickly to the contract award. So the goal here is to help you submit a complete package that's easy to review and accurately reflects your technical approach. Okay. At a high level, there are two primary stages to consider: the solution summary and then the full cost proposal. So for the solution summary, we request a rough order of magnitude, or a ROM, for your effort. This is a concise one-page summary, and a template is provided in the Appendix A. Here, you're going to provide us with a fully burdened direct labor rate, your total estimated labor hours, total subcontractor and consultant costs, and any materials, equipment, travel, and other ODCs at a high level. There's also a resource or cost sharing line there, so please identify clearly in the ROM who is contributing, in what form, and with what approximate value. If you're, for the full proposal, you'll prepare the volume 2, the cost proposal. And for this, you must use the ARPA-H cost proposal spreadsheet. So it's unique to ARPA-H. Some of you may be familiar with this. It's required, and it's required for all of your team members as well. So any subcontractors you propose will also have to use that spreadsheet. So the spreadsheet includes multiple tabs for all relevant cost categories. So please make sure you're going through each tab and completing those tabs, and that rolls up into some total tabs. In addition to the spreadsheet, we require cost and pricing supporting data. There's no specific template for this. It's a written narrative intended to support and provide detail for the cost spreadsheet. So the typical documentation that you would include in this narrative are vendor quotes, any value analyses that you've completed, and other information that justifies all of your proposed costs. So together with the ROM, the detailed cost volume, and the supporting data, all of that helps us to assess the technical merits and the financial realism of your proposal. Okay. So now I'm going to highlight several recommended practices or our do's for cost proposals. So first, do provide all requested information in the cost proposal cover sheet. Incomplete or partially filled cover sheets often lead to delays and follow-up back and forth questions. Please include in this cover sheet a comprehensive estimate of total funds requested from ARPA-H and the amount of any proposed cost share, if applicable. You should also break out in your cost proposal your cost per task. Separate those by fiscal year and phase so that we can understand the time phase distribution of your effort and your spend plan. For any item with a unit cost greater than $10,000, please provide supporting documentation that consists of, we talked about this in your backup documentation, vendor quotes, prior purchase orders or invoices, and information from reputable websites. For your subcontractors, each subcontractor's cost must be broken down at the same level of detail as the prime, as I discussed earlier. So as the prime proposer, you are responsible for assembling and submitting your subcontractor's cost proposal and make sure that they're meeting all of those cost requirements. If your subcontractor wishes to submit directly to ARPA-H, there's a mechanism to do that. Just reach out. And your proposal should also clearly indicate how your subcontractor costs are allocated across your phases and per task. If you plan to use consultants, please provide the consultant agreement or alternative documentation that verifies their loaded daily or hourly rate. And finally, please explain why your proposed costs are realistic given your technical and management approach. Your narrative should demonstrate that the costs are consistent with your statement of work, the level of effort is sufficient to achieve the technical goals and objectives, and that you have a sound understanding of the resources required to execute the proposed work. The linkage between cost, schedule, and technical content is very essential to our evaluation. And I will wrap up with some important don'ts. First, please do not change any of the formulas in the official cost spreadsheet unless it's absolutely necessary to align with your business practices. If changes are unavoidable, verify that the total amount tab and all roll-ups still compute directly and accurately reflect your costs. Compute correctly, rather. Do not delete rows, columns, entire worksheets simply because they're not applicable to your proposal. If you do not need a particular section, you may ignore it, you may hide it. Deleting elements of the workbook can break the embedded formulas and links, and that complicates and delays the review. Do not submit the cost proposal in any format other than Excel. We need the original spreadsheet so that the formulas, links, and structures remain intact for our analysis. And finally, please don't attempt to buy in by artificially lowering your proposed costs. ARPA-H recognizes that excessive emphasis on minimizing costs can push proposers towards a low-risk concept with limited innovation or staffing patterns that rely heavily on junior personnel. We explicitly discourage these strategies. Our focus is on well-justified, ambitious, and appropriately staffed efforts, supported by realistic and transparent cost estimates. So in summary, please use the required templates, provide clear, sufficient supporting documentation, maintain the integrity of the cost proposal spreadsheet, and ensure that your budget is fully aligned. Thank you.
Awesome. Thanks, Sean. I think I want to clarify and also emphasize a few points. Again, we've said it a million times. Please read the ISO. Please make sure you're registered in SAM.gov early. In the BIOGAMI ISO, we are moving away from a traditional prime subcontractor. So that basically means that all members of the multi-party teaming agreement will just need to fill out the budget spreadsheets in sufficient detail. As Sean noted, those can either be submitted as part of the package. There are certain circumstances with business practices where maybe you want to submit your budget directly to the BIOGAMI inbox, and that's okay. Just please let us know so we know to look out for that. Because otherwise it can get lost in the mail and then we deem it non-conforming because we don't have that piece of the budget. Please use a spreadsheet. It really enables us to more effectively review the cost realism of your work. And if you're selected for funding, it dramatically decreases the amount of time that it takes to get under award because we have all the pieces of information there. Okay, so we are running pretty much on schedule, which is awesome. So we are going to take a quick break, and we're going to have then a Q&A panel again from 10:45 to 11:15 a.m. Eastern. So for folks on the line, please make sure that you're dialed back in. For the Q&A, we're only going to be taking questions live in the room. So for folks that are online, we're not going to be manning the chat in real time, but what you can do is you have about 8 minutes. If you want to submit questions in the chat online in the next 8 minutes, please do so. And then my team and I will kind of collate those and we can address them during the Q&A. This is really your time to get any of those burning questions about either the technical parts of BIOGAMI, anything to do with the submission or the review process, as Danielle talked about, or anything to do with the budget. So with that, go forth and mingle. Again, remember the bathrooms are outside the door to the left. There's also water fountains and there's sodas and that sort of thing. And we'll reconvene here at 10:45. We're going to handle Q&As. Let's see. We're going to start with a question that's a little bit more geared towards Danielle. I'm going to do my best to answer it. You can get me if I'm wrong. Okay, so we had a question about multiple submissions. So we understand that you guys are forming teams, and it's possible that one entity, we'll call it Entity A, might potentially want to be part of multiple teams. So the question is, can they do that? Can they submit solution summaries both as an individual as well as part of a different solution summary as part of a team? And the answer is yes. So individuals can submit as members of multiple teams, but they can't be the submitter for multiple teams. So if Entity A is on both Team 1 and Team 2, Entity A could submit for Team 1, but they couldn't be the submitter of record for Team 2. Someone else on that team would have to be the official submitter. Does that make sense? Cool. Let's see, other questions that we got. We had a few questions about TA2 disease targets, and again, about the requirements for neurodegenerative and non-neurodegenerative disease. So I'm going to give a little bit of a step back and explain our rationale for why we made this requirement, both for rare and common, as well as neuro and non-neuro. So as I've said several times, the BIOGAMI program is designed to develop a platform capability that is capable of developing solutions to make intrinsically disordered proteins druggable, regardless of what the indication is. And so we don't want to spend all this time in TA1 developing these models and then kind of only demonstrate the capabilities in TA2 on just one disease target. So why didn't we just let you guys pick whatever disease target you want? We're already being pretty generous and kind of letting you just pick whatever target you want within certain buckets. But we want to demonstrate platform breadth, and so being able to demonstrate modulators of protein misfolding against two separate protein targets. So they can't be just like a wild type protein A and then a mutant of protein A, and that doesn't count as two different protein targets. We also want to really drive towards, again, that ARPA hardness of neurodegenerative disease and not just going for lower hanging fruit that might be more easily translatable to the clinic in cancer or other type of chronic illness. We want to make sure that we're keeping neuro very front and foremost. And so this is why we're requiring two different disease indications, two different proteins of interest, again, one neuro and one non-neuro, and one rare and one common. And you can choose how you want to square that circle, if that makes sense. Let's see. We also had questions about protein targets. Are they considered in scope if the wild type protein is in itself not intrinsically disordered, but a mutation leads to misfolding, aggregation, and disease? This question was already answered on the FAQ webpage, but just to reiterate again, any protein that contains an IDR, whether aggregation prone under normal conditions or solely when mutated, is within scope. Another question that I think maybe hasn't already been addressed on the FAQ online is whether modulators of protein folding could include modifications to post-translational modifications. Yes, that would be considered within scope. And I think those are mainly the questions that we haven't had answered before. So I'm going to open it up to the floor. We have microphones floating around. Please use the microphones. Please also hold it up to your face. This is so the people online can also hear the question.
So this question is also about disease scope, and I was specifically struck in the focus in the ISO around the focus on aggregation diseases. Very much seemed like focus on diseases that are caused by aggregation. In the earlier parts of today, there was the mention of both. I consider those typically are gain of function diseases, but there was a mention this morning also of loss of function diseases. And so I was wondering, is the focus strictly on IDRs and IDPs that lead to aggregates? Sometimes those self-associations are cleared and there's a problem with degradation, for example. So we're strictly focusing on proteins that lead to aggregation?
In this particular program, yes. Now, recognize that there are those other types of situations, but yes, looking and I guess I would also clarify that
because let me elaborate maybe because sometimes a change in conformation will lead to an alternative structure that may be more prone to self-association, but also therefore may be more recognized by the cell's quality control and cleared before it can aggregate.
Yeah, so I guess I would maybe clarify aggregation to include changes in self-association. So phase separation, condensate formation kind of on the path to aggregation. I think that would still be considered within scope, but I would say that the protein target of interest, if that is the case of its biology, that you have to clearly articulate how in the disease indication the protein misfolding and change of conformation leads to the self-association and early stage aggregation.
Thank you.
I saw the answer to the data being open question on the fact, so thank you. My question is, what, if any, requirements are there to make models public, openly accessible, IP-wise, in addition to the data? So just for the model itself?
Yeah, so the models as well, I believe this is also written in the ISO. The models also should be open and publicly available. So the data that goes to train the model, and then the model should also be publicly accessible. Data pertaining, like once the model is then applied to your disease protein of interest, that data can then be considered proprietary as you're moving towards an actual clinical asset. But anything that goes upstream of building the experimental computational models, that data should be open. Does that help?
In terms of models, so the program announcement says things about invertebrate models, and the question I have is, coming out of the administration now, we have, kind of a de-emphasis on vertebrate models, at least at NIH. Is that reflected at ARPA-H or not? I'm wondering if non-human primates are fair game, C. elegans, kind of thinking about the whole range. And
yeah, so I guess I would put this into two different buckets. So I would not consider non-human primates necessarily a high throughput model for TA1, right? I think we can probably all agree that. So we'll kind of put that in a separate bucket. So if we're talking about for evaluating therapeutic efficacy, then yes, that could still be considered fair game. As such, we have no current restrictions or guidance. Now, it is entirely possible that in the 4 years of the BIOGAMI program, there could be other types of restrictions around that. Right. And the beauty of the type of agreements that we set up, these other transactions, that enables us to be very, very flexible with modifications to statements of work. Should new discoveries come up or new restrictions be in place, like we can be very adaptable with teams to overcome those limitations. So in the proposals, just propose to us your best ideas of both the experimental models for TA1 and what you think would be useful for validation in TA2, and just defend those choices. Yeah.
And so kind of a follow-up question, like for the higher throughput invertebrate models, that's going to be allowable and viewed as a reasonable part of a validation funnel?
Correct, yes.
Thank you.
So some of the patients with Down syndrome that, now decade or older develop Alzheimer, will that be considered as well?
So in that particular case, the disease would still be Alzheimer's as opposed to Down syndrome. Yeah.
I have a question about the definition of rare disease. So in many of these diseases, you have a very small subset of genetic mutations that cause similar syndromes. Are those rare diseases, the familial Alzheimer's, the familial Parkinson's, would that be considered a rare disease? Or is that part of Alzheimer's or Parkinson's?
We would still consider that to be bucketed with the broader illness. Yeah. A great question.
Yeah, a quick question about the experimental data that needs to feed into the model. Do we need to have both cellular and the test tube based experiments, or if we have a cellular based like high throughput data, this is enough for this part?
Yeah. So we're just leaving it open to what you think would generate the best data to build the models. So if you feel like your cellular-based system is sufficient to gather all the pieces of information that we would need to address all the questions of biogami, then just let us know that and explain why. Yep.
Hi. I have a general question. My first question is who will be the reviewer panel for the proposals? Because it is both on the commercial aspect and also on the non-profit aspect. So
yeah, so I can't provide specific names, but everyone is a government employee, so they're very often like other ARPA-H program managers. We also have our contractor support staff who are able to provide subject matter expertise. They are not formal reviewers, but they are still bound by the same confidentiality rules. They're considered essentially part of the government team and can help to provide feedback on that.
Follow up on this. Will the data will be shared with the commercial partners regarding the proposals or pre-proposals?
Sorry, can you repeat your question? Are you talking about the proposal stage?
Yes, pre-proposal stage. Will the data will be shared with the commercial partners?
No. So there are no. Okay, so I'll be quite clear that, and maybe Danielle can weigh in. There are no commercial partners that will be evaluating the proposals.
Okay.
Yeah, kind of full stop. So what Char was kind of talking about in terms of engaging commercial partners throughout the lifespan of the program, you'll notice in the ISO we talk about certain types of reporting requirements. So we require, monthly, usually Teams meetings where we get, slide feedback and sometimes narratives. And we also will have annual program review meetings. And so I think one of the questions that came into the FAQ was also where to estimate those in terms of like travel costs. You can estimate that would be like in the Washington, D.C. area. Sometimes we do choose to have them in other locations, but for budgeting purposes, just use D.C. And at those PI review meetings, there are like open door question or open sessions where we can also invite other external stakeholders, like potential commercialization partners that could come in and hear information that you choose to share publicly with them. We can also have closed door sessions where performers share more proprietary data directly with the government without external stakeholders. But for the proposal stage, you don't have to worry about that. It's only government reviewers.
I have a scientific question also. Is there a possibility that intrinsically disorder proteins, which are not directly known in the aggregation-prone diseases, can also be the focus of this program?
Certainly in TA1. For TA2, though, when you're focusing in on developing modulators of aggregation, that needs to be disease-focused.
Thank you.
Yep. Thanks. Sorry, Danielle, was there anything else you wanted to add on the reviewers part?
As federal employees, we are bound by laws to protect proprietary and confidential information. So that, when we receive proposals, we're going to ensure that it can't get out to the masses and share your information. That will be protected. Yeah.
And none of us want to go to jail. I don't think I look very good in an orange jumpsuit. No one wants that. Yeah.
The goal is to work toward transformational therapies. Are there any restrictions on the size of a drug development performer or other parameters?
No, we work with all comers. We're just looking for the best ideas.
One of the critical components of phase separation are the levels of the protein. Would it be within scope to for the therapeutic, maybe TA2, to use a knockdown strategy of the mRNA as opposed to directly targeting the protein?
No, that would be considered outscope because what we're looking for are actual modulators of the protein itself, as well as looking so far upstream at expression. Yeah. But that's a great question. Let me go in the front. I've seen a hand up in the front for a while, actually. First row over here on the right. Thank you. No, you've been waiting?
Thanks. This is getting back to the issue of the IP. So it sounds like there's sort of a division between TA1 and TA2 in terms of what needs to be made public. So just to clarify, if a private entity has a bunch of assets that they've maybe done characterization on, and then that characterization is used to train a model, does that mean that those assets then have to be made public?
Ooh, that is a really good question. And I would say no, that would still be protected. Yeah. But any because also that's coming in, right, as information prior to the BIOGAMI period performance. So any new data that's generated in support of those models would be needing to be open. But if the data is coming in already, then that would still be protected.
Okay. And then even the new data can still be protected if it's part of the TA2 therapeutics.
Correct.
development.
Yes.
Okay, thanks.
Hey, how are you? Thanks for putting this together. For the therapeutic deliverable, is gene therapy or AAV-based vector delivery going to be considered in scope for solutions for this? Okay.
Yeah, we're very agnostic to delivery.
Yeah. Do you consider simulations within the, let's say you have several ways to treat the protein? Do you consider simulation pieces, like AI simulation?
Well, yeah, I mean, that's one of the end goals of TA1 is to try to build toward that capability, right?
And that simulation, let's say that we be at like a bank or potential simulated targets, will that go to you? Is that going to the program or?
Is your question? Are you asking that? So you have the AI silico model, and then any simulations that you generate from that, would those be public? So if they are part of the validation of the TA1 models, then yes. If they are applied specifically to those TA2 assets, then no. That makes sense.
Thank you. So my question is there a preference for validation in invertebrate versus vertebrate animal models?
Are you talking about TA1 or TA2?
Model, like for T1 or TA2?
Yeah, so for TA1, no, as long as it's defended as to why that model and whatever readout you're using from that model would be applicable for validating the particular features of the protein you're wanting to look at, then that is all within scope. For TA2, when we're talking about actual like therapeutic efficacy, we are gearing that towards a vertebrate model.
Last question was, is there a total budget for this and an idea about how many you're going to fund?
No, we don't. ARPA-H doesn't usually announce the total program budget. We will say that multiple awards are anticipated. I recognize that doesn't give y'all a lot of information, but basically, given the ISO and the stipulations that we've put out, propose to us your technical solution and ensure that the budget is actually realistic given the work you're proposing. So again, as Sean mentioned, don't think that you're going to be more competitive because you really lowball it. Like, we've been doing this a really long time. We have a really good sense of, how many FTEs is like really appropriate for the type of work that might be proposed. And so we're just looking for that congruity. I would say that, again, I've been in government. I've been in the ARPA landscape for about 11 years now. I think I can count one finger that I have ever fully funded a proposal with like no red lines to the statement of work or budget ever. It's very, very rare. So one of the beauties of the OTAs and our wonderful agreements officers like Danielle is it gives us a lot of flexibility when we receive the proposals to, hone in on things that we really, really like, we want to make sure we fund, or things that we might feel might have been a weakness of the proposal. And so we might say, okay, like maybe as a total solution, it's not quite what we're looking for, but there's this one nugget that's like really freaking awesome and we want to fund that and see where it goes. We have a lot of flexibility with that. So just be realistic with your budgets, break it out by task. Also, again, as Sean mentioned, that is super, super helpful for us for understanding if we're going to need to make some hard choices about, fully funding proposals or partially funding them. If we know how things are broken out by tasks, it makes our lives a lot easier. So please do that.
2 questions. One, the follow-up question on the gene therapy. Do you consider the CRISPR-based therapy that change DNA sequence as an in-scope, out-of-scope approach?
I would consider that to be out of scope because the premise of the program is to actually be able to modulate protein folding in vivo, and like expression, changing the protein sequence upstream is considered out of scope.
My second question is on the review timeline. So when do we expect to receive whether a solution summary is encouraged or discouraged?
I forget the exact date. It's about I think it's like 9 days. It's about like a week plus like a weekend, because I think the solution summaries are due like on a Wednesday, and I think feedback comes like the following Monday or like a week and a Monday. It's like 9 days.
Thank you.
We will be very busy, don't worry.
Just to follow up on the previous question, so I guess IDPs exist not in a monoculture, but in an environment with lots of other proteins.
Mm-hmm.
So would you consider a CRISPR therapy that targets another protein that influences IDP aggregation to be in scope?
Ooh, it's an interesting question. I hadn't thought about that. I suppose it would. Yeah, because it's still changing something that changes the behavior of the intrinsically disordered protein. So yes, I would consider that in scope. That's an interesting wrinkle.
So another technical question about there's a specific thing for the modeling for 35 amino acid for the IDRs and 100 amino acid for proteins. Some proteins that are aggregating are shorter than hundreds, and they are very important for diseases. And the IDR definition is different depending on which algorithm are we looking at. So these very strict numbers are associated with which algorithm, or with if the protein of interest is aggregating which is shorter than hundreds, should we be focusing on this or not?
So I would say that in terms of TA1 model building, that including shorter proteins are certainly within scope, but we want to make sure that we are still pushing the envelope in terms of incorporating larger proteins into the model. But those can still certainly be included. And if one of the disease indications also happens to be one of those shorter proteins, that's also fine. We just want to make sure that we're not building models solely based on really short IDRs or really small proteins, that we're really exploring the sequence space. Okay, we got like 1 minute left.
Yep. So a follow-up quick then to the review process. So there was details about the solution summaries, but for the full proposal, what is the timeline of the decision on that? And then how much kind of time exists between that decision, the start of the project, and the context for this is potentially hiring staff and stuff like that for projects?
Yeah. So at a high level, we are anticipating kicking off the program, and which means all performers on contract probably by like end of July to mid-August, somewhere in that range. Now, my birthday's in mid-August. That would be a fantastic birthday present for myself to kick off this program around my birthday. So that's kind of what we're targeting. We usually budget between 60 to 90 days for that contract negotiation period, so you can kind of work back from there to have a guesstimate of when feedback would be received. It's always a balance of trying to, obviously I have a lot of personal urgency in trying to get the program launched as quickly as possible without driving Danielle absolutely insane. Want to keep her happy. Yeah, last question.
Sorry, I had a very quick question. So there is mention of dangerous gain of function research. Will proposals be required to disclose anything upfront in the solution summary, or will there need to be just the appendix attached for the full proposal?
Oh, that's actually a really good question, because we mainly framed all of those pieces for the full proposal. However, I would include like a sentence or two in the solution summary. And actually, that might be a good amendment to the ISO, actually, to have an instruction saying like that we anticipate having to address that in a full proposal. So let's make a note of that. Awesome. Well, thanks, everybody. Okay, so that concludes the Q&A. Thank you, Sean and Danielle. This has been a whirlwind of a morning. This program is very near and dear to my heart. I'm so thrilled that it's finally out in the wild and that you guys are all putting the best brains possible towards solving this problem. And I really look forward to seeing all of your solution summaries in about a week and a half. And I guess with that, class dismissed, and we'll see you guys after lunch for lightning talks.